Hello everyone, I'm Priyanka and I'm here with yet another video on colorimetry. This is a colorimeter. As previously we have discussed about the electromagnetic spectrum, as you can see, uh, different electromagnetic radiations are mentioned here from gamma rays, X-rays, ultraviolet rays, infrared to radio waves. Colorimeter is the instrument used for the measurement of colored substances in solution. The instrument is operative in the visible range of the electromagnetic spectrum. Photometry means measurement of light. The color of the light is the function of its wavelength. As the wavelength changes, alteration in color is detected. Colorimetry. It is the most common analytical technique used in biochemical estimation in clinical laboratory. It involves the quantitative estimation of color. A substance to be estimated colorimetrically must be colored, or it should be capable of forming chromogens, that is, colored complex, through the addition of reagent. Colored substances absorb light in relation to their color intensity. The color intensity will be proportional to the concentration of colored substance. The instruments used in this method are colorimeter or photometer or absorptiometers. Let's now talk about the principle of colorimeter. When a monochromatic light passes through a colored solution, some specific wavelength of light are observed, which is related to color intensity. Or we can say that the amount of the light absorbed or transmitted by a color solution is in accordance with the law, that is Beer law and Lambert law. As you can see in the figure at the left hand side, incident ray entered the sample. Absorbing sample of concentration C, and the light that is transmitted. L be the path length of the sample. The measurement of color intensity of a colored solution by photometer is governed by two laws, that is Beer's law and Lambert's law. First we will discuss Beer's law. The concentration of a substance is directly proportional to the amount of light absorbed, or inversely proportional to the logarithm of the transmitted light. That is, A is directly proportional to C. In Beer's law, when a monochromatic light passes through a colored solution, amount of light transmitted decrease exponentially with increase in concentration of colored substance, that is The amount of the light absorbed by a color solution is directly proportional to the concentration of substance in the colored solution. So when we plot a graph between absorbance and concentration, a straight graph is received. While plotting the graph between the concentration and the transmission, a curved graph is received. Let's now talk about Lambert's law. When a ray of monochromatic light passes through a absorbing medium, its intensity decreases exponentially as the length of the light path through light absorbing material increases. So this law is based on the path length. So we can conclude absorbance is directly proportional to the path length, that is L. So we can conclude the amount of the light transmitted decreases exponentially with increase in path length, that is in diameter of a cuvette or thickness of the colored solution through which light passes. That is, the amount of the light absorbed by a colored solution depends on path length of cuvette or thickness or depend of the colored solution. Combining Beer's-Lambert's law is thus expressed as amount of the light transmitted through a colored solution decreases exponentially with increase in concentration of colored solution and increase in concentration of colored solution and increase in the path length of the cuvette or thickness of the colored solution. Photometry measures transmitted light to determine reaction absorbance. Relationship between absorbance and transmittancy. Light falling on a color solution is either absorbed, reflected, or transmitted. I naught is equal to IT plus IA. Here components of colorimetry: tungsten lamp, slit, condensing lens, filter, cuvette, photocell, and galvanometer. Light source will generate polychromatic light, which passes through the slit. After that, a lens is placed, then a filter will only allow monochromatic light to pass through it. Then a cuvette containing a sample is placed. The monochromatic light will pass through it. Then a photocell will detect the amount of light energy entering and convert it into electrical energy. The output is measured by a galvanometer. now talk about the parts of the colorimeter. First we have a light source, tungsten filament lamp. Then a slit. It is adjustable which allow only a beam of a light to pass through. It prevents unwanted or stray light. Then we have a condensing lens. Light after passing through slits fall on condenser lenses which give a parallel beam of light. Next we have filters made up of color glasses. Filters are used for selecting light of narrow wavelength. Filters will absorb light of unwanted wavelength and allow only monochromatic light to pass through. For example, a green filter absorbs all color except green light, which is allowed to pass through. Light transmitted through a green filter has a wavelength from 500 to 560 nanometer. Filter use is always complementary in colors to the color of the solution. Cuvette, that is a sample holder. The monochromatic light from the filter passes through the colored solution placed in a cuvette. It is made up of a special glass or a plastic or quartz material. It may be square, rectangular, round shape with fixed diameter, usually 1 centimeter, and having uniform surface. The colored solution in the cuvette absorbs part of light and remaining is allowed to fall on detector. For example, a solution of red color transmits red light and absorbs the complementary colors green. Next we have detector or photocell. Detectors are photosensitive elements which convert light energy into electrical energy. The electrical signal generated is directly proportional to the intensity of light falling on detector. Then we have output. The electrical signal generated in photocell is measured by galvanometer, which displays percent transmission and optical density. So this is a colorimeter. One for wavelength selection. Second, printer button. At third, concentration factor adjustment. Fourth, UV mode selector, duodenum lamp. Fifth, readout. Sixth, sample compartment. Seventh, zero control. Eighth, sensitivity switch. Relation preparation of solution for investigation. In colorimetric estimation, it is necessary to prepare three solutions: blank, standard, and test. Use of test, standard, and blank. In colorimetric estimation, it is necessary to prepare a blank, a standard, and a test. and the galvanometer will give the optical density that show the percentage of transmission. First we will talk about the blank. Blank is used to eliminate the effect of light absorption by the reagent used. Mostly we use water as a blank, reagent blank. Next we have standard. Standard is a solution of known concentration of the substance. Both absorbance and concentration are known. So the concentration of the unknown can be calculated by using a standard. Next we have test. Test solution is made by treating a specific volume of the test sample with reagent as per the procedure. Application of colorimeter. It is widely used in hospitals and laboratories for estimation of biochemical samples like plasma, serum, CSF, urine, etc. They are also used to test for water quality by screening for chemicals such as chlorine, fluoride, cyanide, dissolved oxygen, iron, molybdenum, zinc, and hydrogen. They are also used to determine the concentration of plant nutrients such as phosphorus, nitrate, and ammonia in soil or hemoglobin in blood, and to identify standardized and quantified drugs. Advantages of colorimeter. It is inexpensive, very well applicable for quantitative analysis of colored compounds, easily transportable. Disadvantages of colorimeter. Cannot be used for colorless compounds. It does not work on ultraviolet and infrared regions. We cannot set specific wavelength as we have to set a range as a parameter. Similar color from interfering substances can produce error in results. Use, care, and preventive maintenance of colorimeter. Read the user manual carefully. Use the correct type of cuvette in the colorimeter as recommended by the manufacturers. Make sure that the cuvette is clean and its optical surfaces are dry and free from finger marks and scratches. Bring filter paper into place before switching on the colorimeter. Before reading the absorbance of a solution, check that it is clear, there is no air bubble in it. Remove the cuvette from the instrument when not in use. Clean the outer surface of the cuvette with tissue paper to remove any mark from the optical surfaces. To prolong the life of the lamp, switch off the colorimeter after use. At the end of the day, disconnect it from the main switch and cover the colorimeter with its protective cover. At regular intervals, check the main power adapter and cable for wear and tears and replace if damaged. Keep it cool places away from corrosive chemicals or fumes. Thank you.